then post on the VIP website. If you have questions at the end of the talk, please post them on the chat or raise your hand and ask the question. So we'll start with Christina Artinto, who is working with Kimberly McAllister. Christina, could you go ahead and share your screen?
Yeah, I'll do that right now. Can you guys see my screen?
Yes.
Okay. I'm sorry, I don't know why it doesn't want to load. Okay. Hi, everyone. My name is Christina Otento, and today I'm presenting how genotyping mice is used to study the role of MHC1.
We aren't able to see any of your slides, though. All we have is a blank screen.
Can you see it now, or do you want me to restart it?
Yeah, you may have to, because we just see a black screen. We are able to see your desktop, but your presentation is black.
Let me see.
Okay, good.
Is it okay if I just zoom in on this?
Yes.
Oh, it's too much. Is that good?
Yeah, that's good.
Okay. Hi everyone. My name is Christina Otento, and today I'm presenting how genotyping mice is used to study the role of MHC1 during synapse formations. Before defining what MHC1 is, I would like to give some background to why the McAllister Lab focuses on it. The McAllister Lab is a neuroscience research laboratory that focuses on cellular and molecular mechanisms of synapse formation during development and disease. We are specifically interested in synapses, which is the location of where two neurons meet, because we know that today in society, there are many individuals affected by neurodevelopmental diseases like autism and schizophrenia. However, in order to develop interventions and treatments, we must understand what happens on a molecular level that causes these neurodevelopmental diseases. Evidence suggests that autism and schizophrenia are linked to immune dysregulation. Given that immune molecules play a vital role in all stages of brain development, a hypothesis emerged that dysregulated central nervous system molecules during development underline the brain abnormalities observed in both neurodevelopmental diseases. That being said, the McAllister Lab specifically studies the role of immune molecules. Major histolytic complex class 1, or MHC1, is the specific immune molecule we are focused on in this lab because MHC1 is a genetic risk factor for autism and schizophrenia. Specifically, MHC1 are molecules found on the surface of all nucleated cells in the body and are composed of 3 alpha subunits and a subunit called beta microglobulin, or beta-2M, which is required for MHC1s to be expressed on the surface of all cells. MHC1 has a role in both the peripheral nervous system and the central nervous system. In the central nervous system, MHC1's role is to regulate brain development and synapse formation and function. Also in the central nervous system, MHC1 may mediate the genetic and environmental contributions to neurodevelopmental diseases. Our lab has found that MHC1 decreases synapse density. Though, what is a synapse? A synapse is a place where two neurons meet to transmit information. This includes a presynaptic and a postsynaptic neuron to transmit information. In this figure, we are comparing a wild-type mice, which contains MHC1, and a beta-2M knockout mice, which does not contain the beta microglobulin subunit and therefore does not have surface MHC1. In figures A and B, the first images are used to identify the pre- and postsynaptic proteins, which then help identify the synapses in the third image. The first image is immunostaining the postsynaptic proteins with an antibody against PSD95, and the second image is immunostaining the presynaptic proteins with an antibody against VGLUT1. The third image then takes each pre- and postsynaptic protein identified to locate the synapses. In figure C, this data represents the synapse density used to compare between the two different mouse lines. Based on this data, we can see that beta-2M knockout mice have a higher synapse density than the wild type, which further proves that MHC1 decreases synapse density. With MHC1 decreasing synapse density, we know that it plays a crucial role in brain development and synapse formation. Therefore, it is important to have MHC1 mouse lines in our lab to be regularly genotyped. The two mouse lines I genotype in lab are called beta-2M knockout and K1D1. Beta-2M knockout refers to the beta microglobulin subunit to be removed from the MHC1 molecule. As a result, that means there is no MHC1 expression on the cell membrane. The next mouse line I genotype in lab is the K1D1, which refers to the genetic locus of MHC1 in mice, as they have two different MHC1s caused by the K and D genes. By having K and D genes removed from mice, it prevents expression of H2K MHC1 and H2D MHC1 in mice. Therefore, there is no MHC1, and we can detect this by genotyping mice. What is genotyping exactly? Genotyping is a process of detecting genetic differences in organisms. There are 3 stages to genotyping. The first stage is DNA extraction, which is when we extract DNA from a mouse to have samples for testing when determining the specific genotype. In doing this, the DNA is digested and breaks open to release DNA. The next step is to do a polymerase chain reaction, or PCR. PCR is the method of making thousands of copies of a single short region of DNA molecule made through a series of reagents and heating and cooling cycles in a thermocycler. The third step is gel electrophoresis. Gel electrophoresis is a technique used to separate DNA molecules according to their size. Once the gel has been in the chamber and the bands have been separated, the gels can be imaged. After gel electrophoresis, we would compare the bands to the controls and determine the genotypes of the samples. This is an image of a gel I had previously run to show the difference between the use of old and new primers in PCR reagents. In a gel, the first column is always the ladder, which is used to compare the band size. Specifically in this example, the controls consist of a wild-type, heterozygous, and knockout mice. The wild-type mice are supposed to have one band at around 470 base pairs. That's that heterozygous has 2 bands, one at 470 and the other at 789 base pairs. In the knockout mice, there is only one band and it's located at 789 base pairs. As we can see in these results, there is a difference in visibility in the 2 sets of primers used to genotype. This is an example of troubleshooting when an experiment does not work out as expected. By using small volumes and many reagents to genotype, and can cause many inconsistent and varying results in our genotyping, though it is something we address quickly. Research is a collaborative effort, and it is important that regular genotyping continues so that experiments can be ongoing and that research can be completed so that more learning experiences can be made day by day about immune molecules like MHC1. There is a community of people impacted by diseases like autism and schizophrenia. And through learning more about MHC1's role in synapse formation, then we are one step closer in understanding the mechanisms of how neurodevelopmental diseases develop on a molecular level. This can then further contribute to the field of neuroscience as we can learn more about the brain's development and processes. Thank you.
Thank you, Christina. Are there any questions? So I have a question on your gel, on your protein gel. So if you could go back to that slide. So is the 219 band that's in the wild type, is that the MHC one because it's not knocked out?
Let me go back to it. These
So
the heterozygous.
your new primers, I'm assuming those are the ones that are giving you better results. So I'm just looking
Yeah.
at your wild type, heterozygous, and the knockout. So it's the 750, I don't know which one, which of these bands refers to what protein?
These are referring to, I think this one's 789
Yeah,
band, and this one's supposed to be 470.
okay. So is the 789 your, I don't know, is there an MHC protein here or not?
No, this is showing the knockout mice, the heterozygous and the wild type. The knockout mice would be referring to the MHC1 not being present.
Okay,
And I guess would mean that it has both genes, so there would still be some MHC1, and the wild type means it does have MHC1.
okay, all right.
Mhm.
Thank you.
Yeah.
All right. Let's move on to our next speaker, Michelle Lopez. She's working with Priyasha. Michelle, go ahead and share your screen.
Okay. Can you see
Yeah.
my
Yeah.
presentation?
Yes.
Looks good. Okay,
Yep.
perfect. Okay. So hello, everyone. Thank you all for being here today. My name is Michelle Lopez, and I've had the pleasure to take part in a project at the Shaw Lab that focuses on the proteomics of ANCL2 variants to discover molecular mechanisms of microcephaly. And so to begin, what is microcephaly? Microcephaly is a condition in which the head or brain is abnormally reduced in size. It can be caused by different factors, including exposure to toxins or pathogens during pregnancy and from genetic mutations. Complications of microcephaly include, but are not limited to, developmental delays, seizures, intellectual delays, and underdeveloped motor skills. There are two main types of microcephaly, but this project focuses on primary microcephaly, which is microcephaly that is present at birth and is a consequence of neurodevelopmental defects. And so at the Shaw Lab, Dr. Fishburne is conducting research that focuses on the molecular mechanisms that induce microcephaly via Zika virus infection, where the Zika virus NS4A protein interacts with ANCL2, promoting Zika virus replication and potentially inhibiting proper brain development. On the other hand, my mentor Nick's project is observing known disease-causing variants of ANCL2 that are associated with microcephaly. So in general, we are more on the genetic side of the focus. And so now the question is, what is ANCL2 and what is its function? So ANCL2 is named after its anchoring repeat and lem domains. It is known as a scaffolding protein, which means it directs the activity of other proteins by acting as a stabilizing platform in order to facilitate protein-protein interactions. So ANCL2 plays diverse roles in cellular processes such as nuclear envelope assembly and disassembly during cell division. It is also involved in regulatory processes, and it also plays many other roles that are still not fully understood. But it has been shown that dysfunction of ANCL2 is linked to improper nuclear morphology, cell division, and human diseases. So the image here illustrates a predicted structure of ANCL2, with the red dots being regions of amino acid substitutions from known disease-causing variants. So a paper by Yamamoto et al. Identified individuals with a type of genetic microcephaly caused by mutations in their ANKL2 gene. They observed a family where the parents were both carriers of variants of ANKL2, which resulted in them having 2 children with microcephaly. So the pedigree here shows an autosomal recessive inheritance of ANKL2 variant, where both parents carried 1 wild type allele, which is indicated by the plus, and 1 variant allele. And their 2 children inherited both versions of ANKL2 variants, which resulted in them having microcephaly. And unfortunately, one of the children died just hours after birth. But it's important to note that this alone does not show that ANKL2 variants cause microcephaly. So to further explore the relationship, researchers looked into the homologue of ANKL2 in Drosophila. The study observed the homologue of human ANKL2 in Drosophila to observe how mutations affected neurodevelopment. And so the control of the study, shown in image I, shows an endogenous version of ANKL2, meaning that there was nothing done to the fly and normal brain development was observed at the 3rd instar larval stage. And so when they mutated ANKL2, they observed a smaller brain size than that of the control. And so to confirm whether ANKL2 is important for neural development, they performed what is known as a rescue experiment, where they express human ANKL2 in the mutant flies. And what they saw was a rescue of lethality and almost completely brought back normal brain size. And so this shows that 1, ANKL2 is involved in neurodevelopment, and 2, its involvement is conserved among 2 distantly related animals. And so now that we have established that ANKL2 is involved in neurodevelopment, let's discuss specific known disease-causing variants that are implicated in microcephaly. And so this image here shows a human ANKL2 where you can see the functional domains, which are the colored boxes, and as well as known pathogenic mutations. And as you can see, there is a mix of both missense mutations and nonsense mutations. And the variants that are highlighted in yellow here are the variants that are included in this project. And so given the relationship between ANKL2 variants and microcephaly, we hypothesized that these disease-causing variants of ANKL2 disrupt protein interactions necessary for normal brain development. And so with the use of proteomics, we can further understand the molecular mechanisms in which these variants either form new interactions or lose some interactions. And then by comparing data analysis of both wild-type ANKL2 and these ANKL2 variants, we might be able to understand the neurodevelopmental pathways in a more robust way and possibly identify what is important for normal neurodevelopment. And so the pipeline of the project can be followed as several steps, where we begin with a Gibson assembly of variant ANKL2 fragments into a linearized backbone with a flag tag at the end. And once we transform bacteria with our plasmid from our Gibson assembly, we then concentrate and purify our plasmids via mini preps. We then transfect hex cells with our plasmid, which allows us to introduce our plasmid with our gene of interest into cells where they can start producing our protein. And so cell lysate is then collected to then perform an affinity purification, which is when our flag tag plays an important role, as it allows us to purify out our ANKL2 variants and their interactors from the cell lysate. And to validate ANKL2 expression, we run a Western blot, and then to validate interactions in our affinity purified sample, we run a silver stain. And immunofluorescence will then help us validate the localization of ANKL2 variants, and the data that is received from mass spectrometry will be used to create an interaction map. And most of my contributions and training was focused on steps 1 through 5. And so far from the MS data, there was about 7,000 proteins detected across all samples. And currently my mentor, Nick, is in the process of analyzing the data and was able to clean down the data down to about 67 confident interactions across all samples. And hopefully we get some interesting insights, but this just goes to show how rigorous the data analysis portion is. And so to bring it all full circle, we began with a hypothesis. We did the experiments necessary to help us explore the hypothesis. And with the data obtained, we can then go back to our hypothesis and make any modifications necessary. So through these mechanisms, the hope is that we get a better understanding of the signaling pathways and regulatory elements that lead to microcephaly. But most importantly, the layout of this project allowed me to experience firsthand the process of the scientific method. And with all that being said, I would love to thank the Shaw Lab for providing an environment that helped me grow as a scientist, and for all the members helping me in all ways, shapes, and forms. And I would love to give a special thanks to my mentor, Nick, for allowing me to take part in this project and guiding me through it all. And I also want to thank Dr. Shaw for giving me this amazing opportunity as an undergrad. Thank you all again, and I will be happy to answer any questions.
Thank you, Michelle. Thank for a nice presentation. Are there any questions? So I have a couple of questions on your transfection. What is the cell you're using?
We're using hex cells.
Okay, and what is your transfecting agent?
The transferring agent.
Transfecting, what is the reagent you're using for transfecting?
So for the
That's okay. I mean, if you don't remember.
we do use some polyjet. We do like a master mix of like polyjet and then our plasmid and we let that incubate for a little bit, and then we place them on the cells. And so that's how we do our transfection.
Okay, great. Thank you.
Mm-hmm.
All right, let's move on to our next presenter, Andrea Rosillo. Andrea, are you ready?
Yes.
Would you share your screen?
Okay. Um, oh. Oh, can you see my screen?
Yeah, it's not in the presentation mode. I think it's oh, there you go.
Okay. Well, hello, my name is Jerosillo. I'm a third-year microbiology major, and I will be presenting on genotyping MHC1 mouse lines to study the role of MHC1 in synapse formation. So as we all know, neurodevelopmental disorders such as schizophrenia and autism spectrum disorder impair affected individuals in their daily lives and impact the landscape of those around them. Thus, it is important to study the mechanisms underlying these disorders to contribute to the prevention and treatment for these disorders. Epidemiological evidence has revealed a link between neurodevelopmental disorders and immune dysregulation. And the immune molecule of interest that is being studied in the McAllister lab is the major histocompatibility complex, also known as MHC1, which is a genetic risk factor for schizophrenia and autism spectrum disorder. And it is also known to negatively regulate synapse density during early development. And due to the key role that immune molecules play throughout brain development, the McAllister lab has hypothesized that the dysregulation of central nervous system cytokines and the receptors are responsible for the brain abnormalities found in both schizophrenia and autism spectrum disorder. And here we can see the gene locus of the MHC1. So H2K and H22 genes are responsible for encoding for the classical MHC1 molecules in mice. And the MHC1 molecules are expressed by all nucleated cells and are encoded by the most polymorphic genes. As for the structure of MHC1, it's a heterotrimeric complex composed of 3 alpha subunits that form a peptide binding group and a subunit called the beta microglobulin unit, also known as the beta 2M. And the presence of beta 2M is required to enable surface expression of the MHC1. And this figure really helps us show the role of MHC1 in synapse formation. So figure A shows an image of a neuron from a wild-type mouse genotype, while figure B shows an image of a neuron from a beta-2M knockout genotype, which are both immunostained by antibodies that recognize BGLA1 and PSD95. For figure A and B, the presynaptic protein BGLA1 is represented by red, while the postsynaptic protein PSD95 is represented by green. The colocalization of these proteins indicates a synapse and is represented by the color yellow. And the graph in figure C measures the effects of the type of mouse genotypes, whether it is wild type or beta-2M knockout, and the level of glutamatergic synapse density. So beta-2M knockout is observed to have an increase of synaptic density of 26% compared to the wild type, and the difference is indicated to be significant. This increase in synaptic density has also been observed in the mouse line K1D1 knockout, and thus to study the role of MHC1 further, we genotype and maintain the MHC1 mouse lines. So the two MHC1 mouse lines that we focus on are beta-2M knockout and K1D1 knockout. A knockout refers to when a gene has been intentionally removed or inactivated so it is not expressed. So the beta-2M knockout does not express surface MHC1 due to the absence of the beta-2M. And the K1D1 knockout does not express surface MHC1 due to the absence of the H2K and H2 genes. And through genotyping, we are able to determine differences in the genetic material of an organism, and it consists of three main steps. The first step is extraction of DNA, in which cells are lysed to release and isolate the DNA. So this is an example of a common procedure that we use in lab to extract the DNA. So first, 1 to 2 millimeters of mouse tail or ear is cut out and placed in the PCR tube, and then lysis buffer is added to the tube and it is incubated in the thermocycler at 98 degrees Celsius for an hour. It is mixed and then neutralization solution is added to the PCR tube, and that's how the DNA template is made. The second step in genotyping is the polymerase chain reaction, also known as PCR, in which the end goal is to amplify copies of a specific DNA segment. And the first step is denaturation, in which the solution containing the DNA template is heated to a high temperature to break hydrogen bonds between DNA bases and promote separation of the DNA into two strands. The second step is annealing, in which the temperature cools down, allowing primers to bind to the single strand of the mouse DNA. And the third step is extension, in which the Taq polymerase attaches to the primers and adds bases to form a complementary DNA strand. And this is repeated until about 60 to 80 copies of the DNA has been made. And the last step of in genotyping is gel electrophoresis, which refers to the process of separation of charged molecules such as DNA based on their size. And this is achieved by running a gel at the desired voltage and time, such as 120 volts for 25 minutes. In this step, the negatively charged DNA travels towards the positively charged electrode. And the less base pairs that DNA has, the faster it takes to travel down the gel. And when the gel has finished its run, it is placed in a gel dock to visualize the DNA bands. And to make sure that the PCR reaction worked, we have to verify the positive controls have the expected results. The positive controls are represented by the wild type, heterozygous, and mutant primers. So the wild type primer usually has 1 band and 261 base pairs. The heterozygous has 2 bands of 410 and 261 base pairs. And the mutant primer has 1 band, usually at 410 base pairs. And many factors can impact the efficiency of the PCR reaction. One such factor is the primer dimer, which is shown by this image. Primer dimer occurs when primers anneal to each other instead of the template DNA, and is characterized by the short length of the bands and bands that are smeared. In the image below, we can see that the first row represents the ladder, which is used as the standard to measure the DNA bands. And you can see that the bands are now well defined, especially for the controls. And to troubleshoot this issue, we can lower the primer concentration, increase denaturation temperature, or increase annealing time. And it's essential to continue genotyping and maintaining mouse lines to contribute towards the research of the role of immune molecules in synapse formation. And studying MHC1 more in depth can help us contribute to treatments that enhance synapse formation to alleviate the burden these neurodevelopmental disorders can cause on the individuals and their loved ones. Oh, that's it.
Are you done?
Yes.
Oh, thank you, Andrea. Are there any questions? So I have one question on your DNA gel. If you can go back to your DNA gel,
Yes.
the last one you showed, the DNA gel with all the.
Yeah.
Yeah. So look at your lane 5, and I'm wondering why you have such a smear.
Oh, so for this gel, we used to make the master mix manually by individually adding every ingredient ourselves. But unfortunately, there was contamination in that master mix with all the ingredients. So now we just order pre-made master mix to
So
prevent.
lane 5, is it one of your knockouts?
Wait, can you repeat that again?
No, the lane 5 DNA sample, is that one of your knockouts?
Um,
Which sample is that? And you got a red arrow for wild type and lane 4, but what is lane 5?
Lane 5. I won't be able to tell because of this gel because it was contaminated, unfortunately.
Oh, okay, all right. I was just curious. Okay, thank you. So we can, we'll move on to the next presentation. So the next presentation is actually going to by both Iman and Elizabeth Moss. Are you both ready? It's going to be a joint presentation from the Alexnard lab.
Yeah, I'm ready.
Okay, share your
Me
screen, and you can do it in any order you wish.
Okay. Okay, I just want to check, is it working?
It's fine.
Okay. All right, so my name's Elizabeth Moss, and I'm here with Aman Shahzad, and we're going to be talking about investigating CHD8 gene function and their association with neurodevelopmental disorders, and we're both undergraduate researchers in the Nord Lab. So the Nord Lab is at the Center for Neuroscience. Whenever I say this, no one ever knows where we're at. We're like all the way over by In-N-Out. But what we do is we work on gene regulation in human neurodevelopment and brain disorders. So we study neurodevelopment with different methods using genetics and genomics paired with neuroscience. And the different systems we use is mouse models and human cells and organoids. So me and Aman primarily work with the mouse models, and we study different diseases, including autism, intellectual disability, epilepsy, and schizophrenia. So our goals are to understand genomic mechanisms of development and disease, and to build different tools and technology for basic and translational applications. So in the Nord Lab, we have 3 different branches of studying gene regulation. So we have the enhancer discovery and functional testing. Me and Aman don't primarily work in this field, but for the monogenetic disease modeling, that's something we're very heavily involved in. We're primarily studying, so that's single mutations in one gene, and we're able to see this in the CHD8 gene mutation that we are primarily focusing on right now. And we also work in environmental disease modeling. For example, in our lab, we have some mice that are exposed to influenza, and we're able to see the different maternal different effects that has on the offspring of that mouse when they're exposed to that, and different mutations that can arise. So our research question is, how does disruption to gene regulation cause neurodevelopmental and psychiatric disorders? So we use the approach of monogenetic mouse models, and we pair genomics and transcriptomics and neuroanatomy to study the function of autism spectrum disorder associated genes during brain development. So like I stated before, we're primarily focusing on CHD8. This is a chromatin helicase DNA binding protein 8, and this gene is responsible for encoding a chromatin remodeling protein. So because of its effects on chromatin formation, you're able to see individuals exhibit autism spectrum disorder when they have mutations in the CHD8. So this graph right here is able to show kind of one of the reasons why we're primarily focusing on CHD8. So as you can see, it's all the way up towards the 16 on the y-axis, and this is showing that it's has a very high penetrance for autism spectrum disorder associated genes. So it's very common for people with autism spectrum disorder, and that's one of the reasons why we are studying it. And we also work with a couple different genes that are listed on this graph too, but we won't really get into that today. So we're able to have models of autism spectrum disorder and intellectual disability pathology in CHD mice. So this is able to be studied through our mutant mouse models that we have in lab. This is done via genome engineering. So in our lab, we are able to have mice who may exhibit the CHD8 mutation, and we're able to study those mice. The way we do that is examining the neuroanatomy, brain structure, and behavior, and then also seeing the molecular genomic and transcriptomic data analysis of the different effects CHD8 can have on these mice. So not all of these 3 branches that we have up here are being studied primarily in lab. We also have different collaborations with the Mind Institute and CNS collaborations where we're able to study all 3 aspects of the different factors of CHD8 and all of the different effects it can have in neurodevelopmental genomics.
Okay, so now I'll be moving into the process of what we do in lab to figure out whether a mice carries is heterozygous for the CHD8 mutation or whether it's wild type and does not carry the mutation. We specifically don't work with the homozygous because the CHD8 mutation is embryonic lethal, which means that most mice that do have it would not be able to survive the embryo phase or would end up at a stillbirth. So how we do this is we end up taking a little piece of tail from the mice, which are numbered so we know what DNA is coming from the mice, and it's taken into the lab. And it goes through an extraction process where it's digested to kind of make it into small molecules and then purified so that what we have is like a clean sample of DNA. That DNA is then nanodropped where we learn the content of DNA that's in it, taking about 1 microliter of sample. The sample can range from 1 to 900, 100 to 300 being about the optimal range of DNA content, which also tells us how much we'll use in the next step. Because if it's on the lower side of DNA, we'll end up using more of it, about 3 to 4 microliters. And if it's in the optimal range, we'll use about 2 microliters. And if it's in the higher range, we'll use about 1 microliter of sample. And now moving on to the next step of our reaction, which is PCR, also known as polymerase chain reaction. And in simple, this is just a process in where DNA is replicated to make a specific DNA is replicated, and this requires a few different things. We have TAC2X4R specific one, which is a DNA polymerase, an enzyme that replicates the DNA, as well as specific primers that we use, which are correlated to the CADH genes, which are put together, and 23 microliters are added to the PCR tubes, including the amount of sample that we'll use based on the DNA content. This is then taken into the thermocycler, which usually has like a file saved, like a CHDA program that runs it. And this is because it changes through temperatures at different times. We start off with a hotter temperature around 95, which is for the denaturing step. And this is pretty much just where the DNA is taken apart to make single strands. Then the temperature is lowered where it's replicated, and this step is gone through a few different times. This can take us about an hour, 20 minutes to 2 hours to happen. After the process of PCR has been run, we'll move it into the gel electrophoresis portion. For the gel electrophoresis is where we figure out whether the mice that we have is heterozygous or wild type. We start by creating an agarose gel. Start by creating agarose gel that is then put into our gel setup, which has a positive and negative side. We place it towards the negative side as DNA is negative, and then we'll travel down to the positive side. And smaller fragments tend to move faster, while larger fragments tend to move slower and will move less. And after the DNA has been placed and we run it for about 35 minutes at 95 microvoltage, we take it to the imaging machine. And after that, we specifically don't really look at how far the DNA has traveled. We look at the bands that show up to figure out whether it's wild type and heterozygous. And as you can see in our gel, the wild type mice do miss the middle band, while the heterozygous have a middle band, which is how we learn whether the CHD8 mutation is part of that mice. And this is really important because the results we have gotten have, the next slide, which leads us to the results, because the mice results that we get can be correlated to humans, because the CHD8 mutation in mice is similar to the CHDA mutation in humans, which tells us a lot about the autism spectrum disorder, ASD, and we have learned a lot about changes in the neurogenesis, which is how by macrocephaly, which is like the enlargement of the brain and the head, which is seen with a lot of people that do have autism, as well as structural changes in the cerebellum. It's a lot smaller. And this is really important because this is helping us learn about the mechanism of ASD and can help with future diagnosis and interventions for ASD. All right, thank you for listening. Do you guys have any questions?
Thank you, Elizabeth and Iman. And are there any questions? I have one question. If you can go back to your DNA gel, I was curious about, yeah, that's the one. So you have the heterozygous and the wild type, but then you have two other lanes where that extra band seems to be even stronger. What are those? If you compare to the HT versus the last two lanes, you got that.
Yeah, so the fourth lane is just another sample, and then the fifth lane is actually our positive. So that's a sample that we know is heterozygous. So that's how we're able to compare and really call the other two samples that are being ran for the first time.
Oh, okay. All right. Are there any other questions? Okay, in that case, let's thank Iman and Elizabeth for an excellent presentation, and we'll move on to the next talk for Dania Tanu. Is Dania ready? Can you share your screen?
Yes, I will. Can you see my screen?
Yeah,
Perfect.
the last slide, I think.
Oh. Okay.
Yeah, good.
So,
Go ahead.
hello everyone. I'm going to be talking about creation of kinesin 1 activating molecule, and I'm affiliated with the Dr. McKenney lab, or we can also call it the MOM lab. And we study a lot of disciplines from molecular, cellular, and biochemistry. Um, and the main lab focus is to study the microtubule cytoskeleton and the motor proteins that are used for transport, specifically the allosteric regulation that follow that movement. And the motor proteins that are studied are kinesins and dienes. And here's a quick background. So what is kinesin? Kinesin is a motor protein that travels along the microtubule to transport cellular cargo. And how does it move? It moves through a series of ATP hydrolysis that would cause their feet to bind tightly and unbind, allowing them to move. And it's this like orange structure over here. And so what activates kinesin? So the MOM lab actually studies this regulation, and there have been previous studies about this. So kinesin 1 has a heavy chain and a light chain, which are strongly inhibited. And in the previous study, we found that kinesin 1 has a synergistic relationship with MAP7, microtubule associated protein 7, and the cargo adapter protein Nesprin 4. MAP7 enhances the mobility of kinesin 1 as it binds to the heavy chain of kinesin, and Nesprin 4 binds to the light chain of kinesin. However, we found that Nesprin 4 has its limitations. So it goes into my research objective. And my goal is to create a chimera protein of MAP7 and kinesin. The chimera protein is a fusion of MAP7 and the active site of Nesprin 2. This chimera protein is hypothetically more efficient at regulating kinesin 1. Previously, two proteins were needed to regulate kinesin with two different binding sites, but theoretically, by combining both proteins and creating only one protein, the regulation should be more efficient. And here's a quick overview of our methods. We first designed the primers. We did a plasmid mini prep. We then followed with a plasmid PCR, a Gibson assembly, then a colony PCR, and a plasmid mini prep as well. And then we did a protein induction and protein isolation. So first, we designed the primers, and we needed 4 primers. 2 were for the MAP7D3 to amplify the backbone, and 2 were for the Nesprin. And we just wanted to amplify the active binding site, which is found in the Lude motif. And the primer is used for both the plasmid PCR as well as the Gibson assembly. So we needed to keep that in mind. And the application that we used to design these primers is through APE. So moving on to the plasmid mini prep, we first started by transforming both of the plasmids with both MAP7 and Nesprin 2 in 10XL gold cells overnight, and then the cells were grown for another day to increase the concentration. A plasmid mini prep was performed using the Zymo research kit, which would extract the desired plasmid. And then we ran and scanned a DNA gel. And as we can see, we have a DNA ladder over here, the Nesprin 2G, and then MAP7. Nesprin 2 showed really low concentrations. And in fact, we did repeat this experiment and we got a higher concentration to move on for the next experiment. And then we did a PCR, which is polymerase chain reaction. And we first started preparing all of the primers and making sure all the concentrations are correct. We mixed solutions of the primers, plasmid, and the template DNA, and we adjusted the annealing temperature and extension before running PCR according to the the length of what we want to amplify. For example, MAP7, since we're amplifying the backbone, it needed a lot longer of an extension time. And then the goal of the PCR is to amplify the regions needed to create the chimera protein. It's just a re-emphasis. And Here is the PCR that DNA gel that we got. And as we can see, map 7, we did split it into two different lanes. However, it doesn't really change that much, as they both showed that at the 700 base pair mark that there was a really clear band, which is a really good sign. And we have a 600 base pair band for the Nesprin 2. And then next we started to extract the plasmid fragments and the backbones. So the way how we did it is by taking this gel and then we placed it on a UV box and accurately cut out the region. And then we removed excess agarose via the Zymo DNA gel cleanup kit. And then we moved on to the Gibson assembly. The Gibson assembly is a technique where a vector, backbone, and a fragment, which is map 7 and Nesprin 2, joined together, creating a new plasmid. And this technique presented with the most challenges, as it needed a lot of troubleshooting. And after several trials of fine-tuning and concentrations of both the vector and fragment, this is what worked. So first, we calculated the concentrations of the vector and fragment to make sure that everything is correct. And then we mixed 3 different ratios of the vector and fragment. So we had a 1-to-1 ratio, 1-to-3 ratio, and 1-to-6 ratio. We ran the Gibson reaction and transformed into 10XL golds and then plated them to see if there are any colonies. And then as a result, we found that the 1-to-3 and 1-to-6 found the best results. The 1-to-1 ratio did not give us any colonies. And then we wanted to make sure that these colonies are correct. So we ran a colony PCR and we created a reagent mixture consisting of the primers, master mix of water, and we picked up each colony and pipetted them into a tube with a reagent mixture. And we then initiated a PCR protocol. And then this is the result of our Gibson DNA gel. Over here, as you can see, on the top, we have the old Gibson, which was a previous trial, and it was really faint bands. And then the 1-to-3 ratio, that was the newer Gibson reaction, it had some bands, however, it had some errors. So then when we looked at the Gibson 1-to-6 ratio, it presented with the best amount of consistency. It was the most consistent. It was at 8,000 base pairs, which is ideal because that is the addition of the backbone and the fragment. And then what we did next, knowing that we got the perfect results, we did a plasmid mini prep to extract the plasmid from the PCR tubes, and then we sent them to sequencing to ensure that the plasmid that we created aligns with the theoretical sequence designed in AVE. And then after getting the results and knowing that we got the proper protein, we did a protein induction and isolation. For protein induction, we first transformed into a cell-specific for protein induction, BL21 P ripple plus APR extras. And then we incubated and grown in LB broth, and then we induced it overnight or with IPTG for 3 hours. And then we extracted that by mini prep. Yes. And then when we moved on to protein isolation, we took the resuspended cells. We resuspended the cells in protein buffer, exonuclease, DTT, protease inhibitor, detergent, NP40, and PMSF. And then using the emulsiflex, we lyse the cells. Emulsiflex is quite a large machine that passes the cells and the solution inside a very pressurized chamber that would essentially lyse the cells without hurting the protein that we want to examine. And then we centrifuged, and then we affinity purified with strep XT beads, and then we washed with a protein buffer. Following with eluting the protein from beads with biotin. And then we followed through with an SDS page gel, and we found that here's the ladder to measure, and then this is a map 7 Nesprin 2 like shortcut. And we can see that overnight and as well as inducing at 37 degrees with IPTG, they both got similar results. So it wasn't that much of a difference. And then knowing that, here are the results. So we wanted to see if there was any proper interaction, and we did it through TIRF microscopy. And then as we spoke before, we said that the kinesin is auto-inhibited, and theoretically, me creating this chimera protein, we can potentially make activation better. And so over here, we can see the microtubule and kinesin together. There isn't much activity. The blue is the microtubule and kinesin is in pink. And over here, we can see long strands of pink, green, and blue. That shows that there is more activity with the chimera protein that we created. And here's a better schematic of how it actually looks. So in green, that's just the chimera protein, and you can see it's saturated the microtubule. And you can see the activity of kinesin moving along that microtubule a lot better than if it was alone without the chimera protein. And future directions, we would like to analyze the landing rate, the velocity, and like the length of the kinesin, how long it takes to go from one end to the other of the microtubule. And I just wanted to give quick acknowledgements to Dr. McKenney, Kyoko Chiba, the Mom Lab, AvenueB, and VIP. Thank you so much. Do you have any questions?
Yeah. Does anybody have any questions? So I have it's really, really exciting results that you are able to actually increase the mobility of your kinesin protein. If you can go back to that slide, what is the source of your microtubule? How are you getting your microtubules?
Oh, we have a solution. I personally was assisted with the TRF microscopy and these techniques. However, I cannot tell you exactly what microtubule exactly we used.
Oh.
Yeah, I apologize. Do you have any other questions?
No, I was really, I mean, that slide you're showing that the mobility of the kinesin.
Yeah.
Essentially, you're able to substantiate your hypothesis.
Exactly.
That's exciting.
I will definitely reach out and find it for you and email you.
Okay, great.
Okay.
Thank you.
Yeah, thank you so much.
All right. So we'll move on to our next presenter, Ramir. Are you ready? Go ahead.
Can you guys see my screen, by the way?
Yes.
Hello, everyone. I am Ramir Tarazi Jr., and I will be presenting the quantification of connexin 36 between beta and delta cells in the pancreatic islet of Langerhans. So our lab, the housing lab, focuses on the pancreas. And within the pancreas or inside the pancreas, there's an islet called islet of Langerhans. We have two different models that we use. We have human and mouse models. We are interested about the behaviors of these different cell types. We have alpha cells, beta cells, and delta cells. Under low blood glucose, we have alpha cells that secretes glucagon to increase our blood glucose level. We have beta cells, when under high glucose, secretes insulin to lower our blood glucose. So these two cell types needs to be regulated. That's when delta cells comes in, but release somatostatin to inhibit alpha cells and beta cells. I also want to mention that beta cells co-release UCN3 or urocortin that binds to delta cells to release somatostatin. So in the world of science, it is well known that beta cells communicates through gap junction, and then the most abundant isoform of this gap junction in beta cell is the connexin 36. So if we look at the mRNA expressions of connexin 36 in these different cell types, we see that beta cells have the highest expression of connexin 36 or GJD2. We have delta cells that has a little bit of expression, and alpha cell has little to none. And these connexin sub-6 subunits form a connexon. And when these two connexons formed together, they form this gap junction. So this gap junction allows us to pass or for ions to flow and some small molecules as well. So as I said earlier, connexin 36 gap junction facilitates the electrical activity and insulin secretion of beta cells. For example, under high glucose, you want this gap junction or connexin 36 gap junction to be activated because we want the intracellular calcium from one cell to flow to the other or neighboring cells to have a higher insulin pool. Because of that, we have like effective it's more like effective on lowering our blood glucose level. Under low glucose, some beta cells are actually active, but we want the gap junction or connexin 36 gap junction to be deactivated because we don't want other cells to be secreting insulin. It can cause hypoglycemia to that individual. So what I'm going to show you is coordination of beta and delta cells under high glucose. So this is a GCAM model that has GFP proteins. So beta and delta cells will be on green. You can see that when they are activated or stimulated, you'll see those intensified green activity. So under high glucose, you can see that some non-nucleated cells are active under high glucose, and then some nucleated cells, which are beta cells, are active as well. Disordered calcium activity within the cells. So under a low glucose, you can see that only non-nucleated cells are active. And this is ghrelin, which is an activator for delta cells. And lastly, we use KCl to depolarize all cell types just to see the viability of these cells. So even though there are some physical attributes or just we can distinguish these different cell types, we want to have a better, identification with these cells. So we use immunohistochemistry, so we use DAPI for the nucleus, and we have GFP, that is the model that we use for this image or for this islet. We have insulin secreting cells, which are beta cells in red, and somatostatin releasing cells with the white. So this is just for the previous recording, we just want to see or identify these cell types. This is an intensity plot with their calcium oscillation or their calcium activity. You can see that below is beta cells. You can see that they have like a cleaner oscillation under high glucose. So that means this is the result of those gap junction, because we said that beta cells communicate through this gap junction, so they have this better oscillation. However, if you look at on the top, this is delta cells. They have oscillation, but it's not as cleaner as those beta cells as well. So in the world of or in the scientific community or in the science community, it is still poorly defined that how these two cell types communicate with one another. So there's like paracrine signaling or gap junction coupling. So our goal for this experiment is to quantify the presence or absence of connexin 36 between these two cell types or neighboring cells. Our hypothesis is that there are some delta cells will have gap junction and are coupled, and then but some that will not. So our method for this is immunohistochemistry. So we use a transgenic mice that expresses MTMG or membrane tomato membrane targeted GFP, but we don't use the GFP part of this experiment. So we only have that membrane tomato that allows us to see that red fluorescent protein in the plasma membrane. So we do perfusion method. So we use saline to flush out those excess blood within the lymphatic system and then the vessels. And we have board PFA to fix the tissue. And then after that, we just harvest the pancreas and do some cryo-sectioning. We use primary antibodies that actually bind to specific antigens. So we have your insulin for beta cells, we have somatostatin for your delta cells, and we have your connexin 36. And we use secondary antibodies. We have to see those different wavelengths. We have blue for beta cells. We have far red or white for delta cells, and then we have green for connexin 36. So this is the image result for that immunohistochemistry. If you look closely, you can see those aggregation between beta cells communication. Beta cells within the membrane, and you have those aggregation as well between delta cells and beta cells. So after we need to quantify these images. So we use CellPost, which is an automated segmentation to annotate this images. So what we do is we draw ROI and we train this CellPost to draw individual ROI. So after drawing those individual ROIs, we subtract them from the original image, and we're just left out with the plasma membrane and those small aggregation. And now we can quantify those results. So what we found out is that there's a significant amount difference between beta cells connexin 36 gap junction and the delta cells compared to delta cells. So we conclude that delta cells have less connexin 36 studying, as you can see in those images, and then delta cells will have a fewer connexin 36 gap junction on average relative to beta cells. So the future direction of this experiment is to quantify an image in a 3D model. So we have this 3D image of a cell or an islet. These we want to quantify in every Z position to have an accurate quantification of connexin 36. So I just want to say thank you to Housing Lab, my PR Mark, Dr. Housing, for letting me stay and guiding me throughout my stay in his lab, and also my grad student advisor, which is Muhammad Hussein Tadi, for letting me be part of his team, and all the members of the Housing Lab and grad students, junior specialists. And I just want to say thank you to AvenueB and VIP for letting me do this.
Thank you, Ramir. Are there questions? So I have a question on your glucose experiment.
Sure.
So you have the low glucose and high glucose. Is the objective here to use osmotic stress?
I'm not familiar with, sorry, let me just go back to that.
It seems to me that when you're varying the glucose level,
yeah, so
you are creating
familiar.
different levels of osmotic stress to observe your
I'm not quite sure with that, what do you call it, wordings, but what I know is that we do have, we use perfusion techniques. So we have this cylinder that allows us to perfuse different concentrations of glucose. So maybe that would be osmotic stress.
I'm just asking because that seems like that's what you're trying to do, but I'm just trying to understand why
So,
higher levels of glucose versus lower level of glucose, you are seeing the differences.
I will get back to you on that and ask more about that.
Okay,
Thank
great.
you for that question.
Thank you. All right, let's move on to the final AvenueB presentation by Tony O. Tony, go ahead and share your screen.
Got you. Do you guys see the presentation?
Yes.
Perfect. Okay. Hi, my name is Tony Vo, and today I'll be talking about how social experience affects cognition in aging voles. Ooh. So before I get started, the first thing I really want to talk about, I really want to introduce the lab that I work at to you guys. I work at the Karen Bales lab, and in this lab, there is a focus on social bonds between monogamous animals. In this lab, we work with 3 specific animals, the TT monkeys, the seahorses, and the prairie voles, which are the animals that I actually work with. Now that we're on the topic of voles, I also wanted to talk about why voles. Why is it so important for us to know about voles? Well, the first reason we look at voles is because they're a really good model species for human behavior because they're so well studied. Fun fact, actually, when I was doing research for this presentation, I actually came across an article that was written around 2-ish months ago that talked about a study that showed that prairie voles showed signs of human-like depression, which is actually really interesting. This shows that they're not only really well studied, but they're also a lot more like humans than we give them credit for. Another reason why we look at voles is because they're both monogamous creatures as well as form pair bonds. Now, a little bit of definition time. What is monogamy? Monogamy is a social construct in which we put emphasis on adult pairs, a pair of adults, versus pair bonds, which is the more psychological aspect of mating. It is the attraction between one mate towards their mate. Another reason we look at voles is because they perform both alloparenting and biparental care. So what is alloparenting? Alloparenting is where a non-parental figure is the one looking after children. An example of this could be grandparents looking after children or siblings. Biparental care is where both the mother and the father have a role in taking care of their offspring. The last reason we look at voles is because they have really fast reproduction. This allows us to study voles in the entirety of their lifespan. On the prairie vole side of the lab, we actually have a huge project that we're working on, known as the Aging Project. The Aging Project's purpose is to examine the effects of the presence and the quality of social bonds on different measures across a lifespan. In this project, we actually split our voles into 4 social groups. The breeder intacts, which in the breeder intacts, our adult pairs are able to reproduce and make children. The breeder vasectomized, where we've actually gotten rid of the male's ability to reproduce, meaning they're not able to have children. The same-sex siblings, and isolated pairs. In this project, we're looking at 3 specific behaviors: the partner preference behavior, separation distress and stress buffering, as well as Barnes maze or cognition, which is what I helped out with. There's also a physiological aspect of this project where we do metabolic testing and other tissue-related measurements as they age. Now that we've talked about the project, let's talk about actual Barnes maze. What is Barnes maze? Barnes maze is a cognition test where we test the spatial memory and learning of our animals. This was actually originally designed for rats and mice, but we wanted to put voles to the test. Now let's talk about the setup. As you can see, a Barnes maze consists of a round table, which is lined by 16 holes. Every single hole will be covered except for one. This hole that is not covered is actually the escape hole, where the vole is actually able to enter in. Surrounding this table are 3 visual aids. These visual aids help the vole to navigate its way to its designated hole. At the bottom of this table lines a fence. For the off chance that the vole were to fall off the table, it'll just ensure that it doesn't run off somewhere. Now, let's talk about some of the information about our actual experiment. The total population size for our vole experiment is 106 voles, 30 of which belong to the breeder intact group, 24 which belong to the breeder vasectomized group, 26 which belong to the same-sex sibling group, 14 of which are male and 12 of which are female, and 26 that belong to the isolated group, 12 of which are male and 14 of which are female. Our experiment is split off into 5 waves, each wave corresponding to different voles. Each wave is split off into 3 rounds, each round corresponding to the age of the vole as well as their stage in the pair bond. Round 1 being around when the voles are around 1 month old before they're paired. Round 2 being when they're around 9 months old when they are paired. And round 3 when they're paired at 18 months. Each round is split off into 10 days, day 1 being the start of the test and day 10 being the end of the test. I also really want to quickly point out day 8, which is actually a really important day in Barnes maze. From days 1 to 7, the vole has one specific hole that I will be navigating to for the entirety of those 7 days. On day 8, we actually flip the table 180 degrees so that the hole is now on the opposite side of the table. We do this so that we can test to see how fast this vole is able to learn where the new hole is. Now that we've talked a little bit about Barnes maze, let's talk about some of the results that we've seen. As you guys can see on this slideshow, I have 4 graphs, each graph depicting time in seconds versus the day that we've done the tests. First, I wanted to look at the female voles. When we look at both the pre-paired female voles versus the post-paired female voles, we actually see that they all show learning as the days go on, all starting off really slow and finding their holes, and as the days continue, they spend less time to find their holes. We also see that, for the most part, the post-paired females do find their holes faster, with the exception of isolated female voles, which is actually the really interesting find in this experiment. As you guys can see, the isolated female voles actually have a lot of trouble finding their holes really quickly. They not only take a very long time to find their holes, but also on day 8, they also take a long time to deal with the new change of day 8, which is really interesting. Now moving on to the male voles. When we compare pre-paired male voles towards the post-paired male voles, we also see that they do also exhibit learning as the days go on, starting off really slow and finding their holes faster as the days continue. Again, we also see that post-paired male voles also are faster at finding their holes as well. But the really interesting part about this is that when we look at the pre-paired male voles, we see that on day 8, they deal with the change of the holes a lot slower than their post-paired counterparts. Just another really interesting thing that we've seen. Now that we have these results, what's next? Our lab is actually really interested in finding the total distance that each vole is traveling in the experiment. For that, we look to Canovia, a motion tracking program that allows us to see the total distance that each vole travels. With Canovia, we're able to semi-automate the process and give us more information than if we were to manually do it ourselves. Not only information in numerically how much they've traveled, but it also allows us to see the patterns in which they travel or just anything of interest that we're in. Anyways, with that, I conclude my presentation, and if there are any questions, I'll be happy to answer them.
Thank you, Tony. Are there any questions? So the difference you see between the male and the female,
Mm-hmm.
I think there is a question from somebody let me see if there is. Yeah, there is a question from Christina. Let's go ahead and, Christina, do you want to go ahead and ask the question?
Yeah, my question is like, why do they go for the hole again? I'm not sure if I missed it.
Gotcha. Okay, let me go back to that slide, the slide of Barnes really quick. So that's actually a really good question. The reason why these voles actually actively find these holes is because voles are a very prey species. They're the yummy snacks of a lot of animals out there. So because of this, when voles are like outside in open spaces where there's bright lighting and all that, their survival instinct is to go hide inside like a safe, closed off area. So that's why our voles actually go find the holes themselves.
Thank you.
So
Of
the
course.
follow-up to that is why is there a difference between the males and the females?
The males and the females. What do you mean by your question?
So you said that for example, I'm looking at your frame 2, the female voles. I think the second graph is the one that were isolated. I can say 6 months post-parent.
Yeah.
And the top graph, which is what the different groups you got. So I'm looking at that on your next frame, which is the male, and there seems to be this when you switch I guess
the holes
from the 180-degree switch, the ones that seem to be having the most trouble finding the hole again seems to be the one on the top in both cases. I don't know what they are. So that's what, yeah, that one and the next one. The
gotcha,
yellow
this one as well.
Yeah, right.
Gotcha. So at the moment, since this is only our preliminary results, we're actually still in the middle of like investigating why everything is happening. At the moment, we only have like the time of which they took to like actually finish the test. I think right now we're also trying to, because I know they are also like taking a long time. The ones that are taking a long time, we're still trying to see like patterns that they follow or like just certain behaviors that they look at to like see why they're actually like having trouble finding their hole.
So the follow-up question to that is because it's also following the trajectory of their motion, right? Because now you have a way of actually tracking like you're showing here.
Yeah.
Or the ones that are taking the longer time, are they kind of wandering around before they find it?
Hmm. Yeah. So what we see is that for the ones that take really long time, we see that they're not the ones who are going straight to the hole. They're the ones that are like they're like either systematically going to every single hole to try to find their hole, or they're like very stressed. They're like running around as much as they can.
Yeah, okay.
Hmm.
Thank you. There are any more questions? Then let's thank all our Avenue B presenters for excellent presentations both in the morning and the afternoon. And let's see, do we have our Avenue E presenters?
Yes, I'm here.
Okay, Yusuf, go ahead. Share your screen and we are ready.
Right. Sorry, just one sec. Oh, I'm sorry, I'm having problems with my computer for some reason.
You need to reboot, it's okay.
Yeah, all right.
Go ahead,
One
go
sec.
ahead and
Sorry
reboot.
about that.
No, we can wait, no problem.
All right, sorry about that.
Ah, there you go.
Can you see my screen?
Yep,
All right.
you're good.
Okay, so hi everyone. My name is Yusuf Muchos, and I'm going to share my experience as a research assistant at the Cyberhuman Physical Systems Lab, or also known as the CHPS Lab. So a little about myself is I was a mechatronics engineering student at the German University in Cairo, and I transferred to UC Davis as a mechanical engineering student in 2022. And I would say my top two interests would be robotics and computer vision, and that's why I jumped on the opportunity to work at the CHPS lab on the Predictive Intelligence for Pandemic Prevention project, and I'll be talking about that in a sec. Of course, I wouldn't be able to do that without the support of AvenueE and the LAP programs. So a little about the CHPS lab is many topics of study include control theory, artificial intelligence, formal methods, and cognitive science. So also research focus areas are in fields like adaptive human autonomy teaming or cognitive neuroengineering or UAV swarms or UAV drones. And these have a variety of applications in areas like agriculture, environmental, public health, and defense. I've worked closely with Professor Gong, who's a professor at the MEE department, and also he's the director and PI for the CHPS lab, of course. And I also assisted the PhD student Behan during this project as my job as a research assistant at the lab. So, what is the Predictive Intelligence for Pandemic Prevention Project, or also known as the PIPP project? So, it has three main ideas behind the project. First phase would be habitat observation. So, basically, we utilize technology to conduct surveys of bat caves to monitor bat behavior and population dynamics, which helps us identify potential disease hotspots and track changes over time using different sensors we have. Of course, this is inspired by the COVID-19 outbreak, and this is our attempt to try and minimize the chance of this happening again through the constant surveying of these bat caves, looking for another disease that might come up. Second stage is air sampling. So we use an air sampler to basically collect air samples from these bat caves Then we get back to the lab and analyze for pathogens in order to provide early warning signs of potential threats. And last stage is basically integrating the data from our sensors with the air samples we collect in order to predict, develop a model that can predict these future pandemics. So this is the air sample we used. It uses a microfabricated gas P concentrator in order to collect VOCs, or volatile organic compounds, from site, and we get it back to the lab to analyze. Another component we use is the LiDAR sensor that can map areas using light lasers up to a range of 100 meters. So it's really useful, especially in bat caves. And of course, we use also a thermal camera that helps us see, because most of these bat caves are dark, so we cannot introduce any source of light in fear of being intrusive to the environment. So a thermal camera helps us identify hotspot areas in the cave. So first phase was that we built a handheld device, which is this one, that includes the LiDAR sensor, the VOC sampler, and the thermal camera, with also a screen and a small computer that helps us to map the areas while like walking in it through a cave. So basically tested that as we see in the hallway of the academic search building, and we can see that the LiDAR was able to pick up like hotspots around the hallway, and the thermal camera also worked as expected. So the second step of this project was to mount this device on a drone in order to be able to fly it maybe autonomously inside the bat caves. And the reason why we want to do that is because a handheld device needs to be operated manually by someone who has to go into the cave and out of it, which increases the risk of contamination and becomes prone to human errors, and is a bit invasive to send people inside these caves also. So all that would be eliminated with the drone. So the first step of designing the drone was to decide between quadcopter drone and hexcopter drones, which is basically just deciding if we're going to use 4 motors for the drone or 6 for it. So this is comes down to basic weight analysis. So we weigh every component we have and get rough estimates if we decide to add cables to it maybe. And then we calculate each weight of the quadcopter and how much weight we would need, or would the final weight be using the 4 motors. And we do the same thing for the hexcopter components, and we weigh how much the final drone would be with using 6 motors. And then we finally analyze the thrust and calculate the thrust calculations we need for each drone, and we decide which one would be more efficient, which design would be more efficient to go with. So in our case, we decided to go with the Hexa drone because our components are fairly heavy and we need the thrust from 6 motors to carry it. And as you can see, we also designed a propeller cage in order to protect the bats from being hit with the propellers of the drone for their safety. And also, we decided to bag the entire drone components with a polyethylene vacuum bagging film that is sealed with yellow sealant tape. As I'm not sure if you can see it, but the bagging goes around the main components of the drone except the propellers in order to reduce the risk of contamination. So far we were able to build the handheld device and design the drone using 3D CAD, and we actually assembled some of the motors and propellers onto the drone. And in the future, we're planning on 3D printing the mounting brackets in order to put all the components on the drone themselves and fully assemble the drone and do a lot of testing, especially flight testing. Thank you so much. Any questions?
Thank you, Yusuf. Are there any questions? I have a question. So
Sure.
when you come to the testing stage, what is it you plan to do? Do you want to use in a simulated environment to see if you can actually fly them and test the samples?
Right. So we would basically do a similar thing like we did with the handheld device over here. So we actually used hand warmers that we attached to the walls that would simulate the heat signature coming off from a bat. So and we also have a special built mesh net in the CHPS lab. I don't have a picture of it here, unfortunately, which is just like a huge net with it like a safe area for the drone to fly in.
Okay.
So this is how we mainly plan to on testing it.
But how do you simulate something like a bat? In other words, one of the things you mentioned was that these are to be able to fly through caves, but not essentially disturb the bats or kill them or what have you.
Right. So we have no idea how we're gonna test it through a cave right now, to be honest. But we have so far simulated having a bat as equivalent as having a hand warmer just placed
Okay.
on the wall, giving off like a heat signature.
Yeah.
So and for the drone, we're gonna test like the flight of the drone in the lab using mesh networks just for safe flight because the drone is pretty huge and we cannot test it normally in like without a license and a safe place for testing. So that was the thing that we're worried about now. But in the future, we're gonna have to, of course, test it in actual bat caves. But we're working on that right now.
I was wondering whether you could use something like moths or something, instead of bats, you could moths. You may be able to collaborate with some entomology lab and get some moths.
Well, it's an idea.
Just wanted
Yeah,
to.
I might actually tell some professor about it. But yeah, it's a good idea actually.
The moths are cheap. You can kill them if you want, but
Yeah, I
okay.
guess that was one of my questions was, would you, in a test environment, need any permits or something like that to actually fly this into a
Yeah,
known,
yeah.
habitat?
We actually need license like a licensed drone pilot to fly our drones, which they have to go through testing and take an exam in order to be up because the drones are like commercial drones, so they are pretty huge and use a lot of force and thrust, not like just normal like small drones. So they actually need a license to fly it, and of course we need all sorts of licenses to be able to go into a bat cave actually and test it.
Yeah, and we've mounted those FLIR cameras, the IR camera that you showed there here at Chevron.
Cameras? Oh,
Yeah, so and those are in grams, right? All the weights that you had for each of the
yeah,
components.
it's in grams. Like this is 92 grams for the thermal camera.
Great.
Yeah.
Good work. Thank you.
Thank you so much.
Are there any other questions? Let's move on to our next speaker. Hongui Lee, are you here? Oh, seems like you're missing a speaker. All right, let's move on to the next person. Foram, are you ready?
Yes, I am. Let me just
Go
pull
ahead.
my presentation really quick. 1 second.
Do you know what happened to Hongui? Because he's also from your group.
He's not from my specific group. I think he's with the same professor, but I don't recall if I haven't heard anything, so I'm sorry.
Okay.
Yeah.
Because he didn't inform me of that he won't be here.
Oh, I see. Yeah, I'm not too sure, unfortunately, but I can pull up my presentation really quickly.
Go ahead.
Let me, yeah. Sorry. Okay. Oh, sorry. Okay. Can you guys see my screen?
Yes.
Perfect. Hi, everyone. My name is Forum Shah. I will be presenting on behalf of my group on data magic, building ML tools. I was working under the mentorship of Professor Rafatirad, so I'm really excited to be here today. Thank you so much for your time. So um, when creating a research project, we wanted to really focus on the issue of equity. We're all CS majors as part of my teammates and I for this project. And we realized through our research that there's a huge equity gap in terms of AI and ML understanding, not only in the workforce, but also with different age groups out there, different minorities and different gender gaps. With some studies that we found, we saw that 84% of people on a daily basis don't realize that they are interacting with AI and ML on a daily basis. And there's unequal, like I mentioned, gender, age, and minority representation in CS, which leads to a large knowledge gap. This is extremely distressing because AI and ML are on the forefront of a lot of major sectors out there. When we looked at the scope and the business model and benefits of them, it's used in many sectors like finance, business, genetics and genomics, healthcare, retail, education, and a lot more. So as it quickly becomes the forefront of a lot of different businesses out there, and it's grown over 270% in the last 4 years, we wanted to create a tool that would really educate and teach people on the basics of what AI and ML do, and more importantly, how it helps with automation, collaboration, and clarity and efficiency, because it really is a great tool, but people need to be able to know how to use it in the first place. So in order to accomplish that, we just wanted to first really cover what is machine learning in the first place. So machine learning is a subset of artificial intelligence. It has the capability of a machine to imitate intelligent human behavior. Basically, it's just software that you create that can make predictions based on data sets that you give it that would kind of replicate what a human would also find from the similar data sets. And we wanted to create this tool to educate people on this in order to kind of show them how data cleaning with ML can create a lot of good benefits out there in order to promote a lot of awareness. So when doing current market research, we also found there's a lot of other tools out there like TensorFlow, OpenRefine, and DataPrep that offer a similar open source environment that allows people to understand and generate knowledge on what ML is and allows them to do data cleaning. But and it also allows them to do a lot of powerful resources, such as detect and remove duplicate data. If data sets contain duplicate data from one source, it can often skew the data set. So they have tools where you can remove that information, which is really helpful. It also helps with scalability. If you're using a large data set or want to merge different data sets together, they often have tools where you can really nicely be able to look through it and skew it correctly. And it also, like you see here, it helps with data profiling and data transformation. But unfortunately, we found in all the models that we found in all the open source platforms out there, you really need a CS background to really see the nitty gritty and understand what you're doing. With our goal of it being to educate and inform people of what this is, it doesn't really help us fit our goal. So there's a definite gap out there. And that's how we kind of found that this is what we wanted to target. And this is the trajectory that we wanted to take for our project, and kind of created our own idea to create a user-friendly platform where people can not only learn what machine learning is, but also see visual examples of how it works, rather than just looking at code that a lot of people wouldn't understand what it means a lot of the time. So in order to kind of go about this process, we decided to split up our learning and our understanding into 4 distinct modules. We wanted to inform people on what pre-processing, supervised learning, unsupervised learning, and reinforcement learning are. So just really briefly, pre-processing is basically the first step of data cleaning. It focuses on reanalyzing collected data to kind of correct any errors before it goes through the data correction process. And basically, you're just gaining information from the data itself. All the commands I mentioned earlier, like data deduplication, data transformation, it handles a lot of those steps right at the beginning just to make sure that the future steps work on accurate data. Then there's supervised learning, where you can train models from specific data sets to build on its own knowledge. For example, there's classification and regression. With supervised learning, basically you're giving it labeled data for it to make predictions on what it should look like or how accurate it is based on the data that it's given. And then there's also unsupervised learning. So unsupervised learning is when you're training models on unlabeled data sets. So it's trying to see what patterns the algorithm can find without having specific instances or things to look out for that are given to it already. And this really helps with finding underlying patterns or kind of I guess, patterns and other things that you can find without knowing it, just looking at it firsthand. And a really cool example of this is clustering that you can learn about, because it's a really cool algorithm for how to group together information that you wouldn't have learned about at the first place. And lastly, there's also reinforcement learning. So reinforcement learning is really important because it's a linear sequential model that helps make predictions based on past data it's collected in its own environment. But it's especially useful because it doesn't just use one single data set or just one single platform. It uses a collection of data sets to make different predictions based on its own environment through passes and failures. So it's a really good example of building on its own knowledge as well, because it uses its own data continuously to make sure it's being able to progress further. So these are our 4 modules that we wanted to focus on when creating our educational platform of a website. For each of these modules, we decided to create different algorithms to kind of showcase visually what these different platforms look like when actually reading, because you can say this means clustering, but you don't know what it actually means until you actually see it in action. For example, I worked on an algorithm called the KNN algorithm, which stands for K nearest neighbor. This is an example of supervised learning because basically the algorithm is learning based on the labeled data that you give it. For this algorithm, I worked on the iris data set from Python. And basically, based on the data that you give it and the classifications that you want to make, it will check based on the data points that you have. For example, here it's iris, so they're flowers, different data points of flowers. Based on what you want to classify it by, like for example, petal length and petal width, it will then, based on what its neighbors are describing as the correct value, it'll then make its predictions based on that. And based on what you want to classify it by, it'll then output a really nice data plot to see what that correlation looks like based on the 2 specifications that you give it. This is really helpful to try to understand how different categories can make different predictions and how to see it visually, how this all kind of comes to light. It can ask the user what they want to classify by and then output a really nice data table as you see here. And I also worked on a feature engineering kind of platform, which allowed you to learn more about pre-processing and correcting data before it was fully ready to go for the future steps. So what does our platform actually look like? So this is kind of our basis for what our website looks like. When you first open it, you kind of see a welcome page. As you can see here, it says welcome to Data Magic. And right here, we wanted to really emphasize the importance of visual understanding. So we have a collection of different videos that people can learn from and kind of gain insights about. For example, there's ML basics, ML models, ML versus deep learning, and supervised versus unsupervised learning. These are all kind of embedded into the website. So as soon as you click on them, you're able to be taken to some really helpful videos that we researched and found really applicable to what we're trying to teach. And you can kind of filter through them and also like add more videos if you want to as well. Well, this is a never ending kind of process. There's a lot of helpful things out there, but it's kind of our basic model for how it should look like at the beginning. And then when you continue to scroll down, as I mentioned earlier, we wanted to focus on the 4 main topics. We don't want to overwhelm anyone right away. So we just want to delve into the 4 overarching topics of ML, that being pre-processing, supervised learning, unsupervised learning, and reinforcement learning. And when you click on any of these modules, it'll take you to a separate page, which kind of tells you more about what that sort of subset of machine learning is, and more importantly, what algorithm that you can look at and how that algorithm works. What are the key topics of that algorithm? And as you see at the bottom of the screen here, there's this visualize button, which is visualize the algorithm. And when you were to click it, it should take you to something similar to this, where you're able to interact directly with how that algorithm works and what it looks like, without having to worry specifically about the backend code or how to code that specifically into your terminal or anything. All the hard work is already done for you. You just get to decide how you want to see it, how you want to view it, and how you want to learn about it, basically. Sorry. And basically that really helps with the summary of it all. It's a really great way to kind of allow for a more personalized learning experience. Throughout this journey, we thought it was very important to learn about not only what the topics are, but how you learn about them. And visual learning and understanding is a key component to a lot of computer science. So being able to portray information in this way, we thought was really helpful based on the research that we did and the overall consensus that we came to as a group as well. And like I mentioned, the hands-on learning mechanism is really crucial too. You're not only seeing through videos how it works, but you can actually actually play around with it yourself through the interactive buttons and interactive features on the website, which is really helpful. And it's more complete and a holistic learning content rather than just showing you what it is and going on from there. We really take you through the modular process of step 1, 2, 3, and 4 to help you understand the overall topic of what machine learning is and how we get to accomplish that on a daily basis. So we really hope to promote equity and understanding through this project, and I'm really grateful for the experience of it all. So thank you so much for the opportunity, and to my teammates and to Professor Rafatirad for the great opportunity to learn research along the side. It was a really great opportunity, so thank you.
Thank you, Forum. It's really exciting work. Are there questions? Yes. Somebody? Shannon, do you have a question or? Okay.
I have a question for him. Who's the target audience for this?
Yes.
College?
I'm so sorry. Continue.
Go ahead.
Oh yeah, so that was actually one of our main questions when we were going about this. So our target platform is not, we do want to target college students, but it's more based on for actually people who are not in school. So more like people of an older generation or people who are not currently interacting with these topics or lessons through school or anything. So we were targeting more of like of an older generation or people who are not in school at the moment, because oftentimes at least as CS students, we can learn these through classrooms, but they don't have the opportunity to learn these the same way. A lot of classes and platforms out there charge a lot for the education, so we wanted to make this a free platform to accommodate that gap.
And then
I have
a
a
follow-up to that.
Oh, go ahead.
Go, Yasina, you go ahead.
All right, thanks, Matt. I just have a follow-up. I was thinking about sort of when you started introducing the topic, you were talking about wanting to address the disparities in participation in STEM, right, or in computer science specifically. And so I'm just wondering if in the future, perhaps you see an evolution of this to target middle school or high school students, because it could be an opportunity to introduce them to the field and then potentially, create more computer scientists.
Yeah, definitely. I think that's definitely a goal that we have for the future. I think reaching any audience in itself would be an amazing accomplishment. I think as we continue to grow about it, going to the schools and being able to, I think, make it even more interactive for younger audiences would be great, to just kind of capture their attention a bit more and teach them what it is. Because at least when I was in school, we didn't have all these topics to learn about. So being able to make it more interactive, I think would be a great way to get them engaged as well.
There was another person.
Yeah, and I was just going to add to that kind of same line of questioning. What kind of groups did you involve in testing this out at all?
So in terms of the actual testing process, we're still in the process of fine-tuning the website a bit. So we're a bit far from actually testing it, but we hope to cater to it to different age ranges to try to see which age range it kind of specifically love it and which ones don't love it, just to be able to figure out and fine-tune it a bit more. So we're hoping to, I think, reach out to local schools in the area as well as older college students to kind of gauge that range of where we can improve, where we can do better, but we're trying to fine-tune a little bit more of the details before we reach that point a little bit.
Sounds good. Thank you.
Okay, there are any more questions? Thank you, Forum. Let's move on to Hungui Lee. Go ahead and share your screen.
Yeah, sure. Can you hear me fine? Hello?
Yeah, we can hear you.
Okay, let me share my screen. Oh, wait, let me put it in here so I'm looking at you guys. Oh, that cancel. You share. Screen one. Okay. Can you guys see it?
Yeah, we can see it.
Okay. My research topic is large language model in hardware vulnerability mapping, and it's also supervised by Dr. Ravatira. So a little bit background about my research. Hardware is a specific vulnerability related to to the hardware design language, which involving the design flow of a specific hardware, and it related to the vulnerability can occur in the design flow, and you can have some malicious injection in the design. and can cause other security issues. And why does it matter? So to prevent the hardware vulnerability is crucially important because first of all the software is made on top of the hardware. And if there's vulnerability in the hardware, it's gonna cause issue in the entire system. And a little bit highlight on this slide. The most important thing about this research is if we're able to detect the vulnerability from the designing phase, is the most cost-effective way to dealing with this type of security issue. So we can prevent this hazard before it happens. And the other thing why we pick hardware as our platform is there's a lot of research done on the software vulnerability detection, but not as much as in the hardware vulnerability. First of all, because there's a lot less data in the hardware vulnerability area, and I think it's a bit different than the software, and the code amount is significantly less due to software just a lot more general and a lot more applicable. So the technology we use is a transformer, which is a fundamental underlying technology for GPT and all the large language models nowadays. So, the transformer model is being introduced by the paper Attention is All You Need 2017, and it relies on its own attention mechanism to simplify the language processing process and make it more robust and more powerful. And also, and nowadays we found that transformers can be used not only just on text, it can be used in image, speech, and structured data, and a lot of other various subjects. So it's very robust techniques and very powerful techniques. So some other advantage about transformer is the key thing is transformer is able to be parallel trained. So the previous model on natural language processing problem is mostly recurrent model, and their states of the computation depends on the previous state. For example, meaning if I'm trying to do some processing on a paragraph, if I need to get a representation of the entire paragraph, which is include the last word, you'll need a representation of the previous word, and which needs a previous word. So that means if I want the computation of an entire paragraph, I need to go through n computation to get that final representation. Instead, in transformer, all this computation can be separated by themselves, so they can be computed individually, which allows to the actual training process to be distributed to multiple GPUs, multiple computation units to be done in parallel, so which is much faster and much more scalable. And related to the attention mechanism they introduce, transformers yield a better performance on the complex natural language processing tasks. Another good thing about a transformer is we can use a pre-trained model and build on to the transformers, build on to the models some company already trained it and are there available for us. Okay, let me bring up. So if you can see from this example, if we have a foundational model, we can do our customized fine tuning on top of it to make it adapt to our specific subtext in the subfield. And okay, so for our research, our data sets mainly the common vulnerability enumeration, common weakness enumeration, and common attack pattern enumeration and classification data set for MITRE website, which is a non-profit organization that collects this security-related data. And this data set contains both software and hardware vulnerabilities, and our research use mainly the hardware vulnerabilities, but we also use the software as a not add-on supplement for a data set. So we have a better understanding between the model have a better understanding between the relationship between the vulnerabilities by learning the softwares, and hopefully it will transfer to the hardware vulnerabilities. So our work includes a fine-tuning of a large language model, and we pick the model that's published by Meta, which is called Llama. And on the data set we collected to make it adapt to this the work of mapping the links between power vulnerabilities. And we also attempt to generate example code for each Verilog, which is a Howard design language, applicable CWE weakness eliminations. So attempt to gather this data set for future research. So we will have an example code of what each vulnerability looks like instead of just their documentation of how they behaves. So some examples of our code. This is a fine-tuning process we have, which we take the data set, we do the pre-processing, and we gather the base model from BERT and train it to compile a local model which we can run to ask question to it. And before we do the training process, we had to clean up our data and do a data augmentation. As I mentioned before, the vulnerability elements related to hardware is a lot smaller than the software. So we have to do some data augmentation tests to make sure that we have sufficient enough data, and also to clean up the data distribution on each label. So what we do is we, first of all, limit the amount of the most large label to 400, so it's in a more close ratio to other labels. And we also mirrored some of the label that's one way. For example, if A is a parent of B, we generate a label of P is parent, A is a child of B, we generate a label for B is a parent of A. So we can mirror that reverse relationship in our training as well. This is just a training process, and the loss, the scoring, the performance increase towards the training set. And as a result to our, this is a visualization of the result, which is a true label versus a predicted label. If that means the diagonal is the correct prediction, and the other is what it's supposed to be and what is being predicted as. So those are the false predictions. And some other work we did is to benchmark the model, because once we train the model, we want to know how good it is after the training, how much improvement it got from the training. So we perform some model benchmarking before the training and after the training. So this is the benchmarking for, this is actually the benchmarking for the model with different parameter size, which the more parameter it is, the more detail and the more knowledge a model kind of can learn. And these are all the pre-trained model we have. So we do the benchmarking on this base model to see how much performance gain from the increase of the parameter size. How this benchmarking is done is that you'll give each model a prompt, ask it to generate the Verilog code based on this the inputted command and the description of the function. There's 3 levels of the detail of the descriptions, which and then the model will generate the code for this this prompt. And we take this prompt, compile it, and run the unit test to see if it passed what we expected, or if it failed to generate a correct code on our prompt. This is just, we have 13 example tests, and each test is being done in different temperature. A temperature is a parameter that affects how aggressive a model is in guessing the result. So the higher the temperature, the more randomness in the generation. The lower the temperature, the more it will only output the most confident result to us. This is an example of our generation for CWE20, which is include the data encryption. It's not encrypted. So we pass the prompts in and generated a code like this, which it includes this vulnerability, which it did not encrypt the incoming data. Or it doesn't verify the data. It doesn't verify the data, so it satisfied our prompt, which to ask it to generate code, including this vulnerability. The other work we did is we tried to verify the code we generated. So after we generate the code database, we aim to verify to see if these codes are the correctness of this code, and also if it contains the behavior of our intended vulnerability. Okay, so the previous test is a static test. It compiles. Wait, it's not. It's a correct. No, it's just to test if this code is runnable and if it fits the. Wait, sorry. So the previous test is test on the model's code generation ability, and this test is testing on if the code generated fits the prompt we need, which contains the vulnerable behavior. Because we're not able to manually verify all the lists, we built this verification tool to ask the model to analyze the code, and we compared the analyzation against the original description to see how similar they are, to determine if this is a good entry for our data set. It's not. Oh, that's pretty much the work we did, and thank you for listening.
Thank you, Hungry. Are there any questions? So I have a question. You mentioned initially that you're using the transformer model, which is the basis for things like ChatGPT.
Yes.
So my question then is, when you're talking about the temperature, the aggressive versus the less aggressive,
yes,
is the hallucinations that you see with these models because of the aggressiveness at which?
Can you repeat that? Sorry.
So, for example, with ChatGPT, when you depending on the query you provide, you can get, completely made up answers, hallucinations. So, is that because of the temperature effect that you talked about?
Yes, but it's not the main reason. So let me pop. The other thing about transformer is guessing the token based on what is seen. So that means whatever is generated from model from like ChatGPT is not the actual fact. It's not something it knows. It's generating token by token. You can think as word by word to fit like what the previous is being inputted. For example, if you input I like and leave it empty, you'll guess the best word is seen before what it can be. So it might be a fruit, it might be anything. So that means the result from GPT is guessing. It's all guessing.
Okay. All right. Somebody else? Okay. I was trying to check the chat box to see if there's any other questions. Are there any other questions? Okay, in that case, let's thank all our presenters and for a very exciting research you're all doing. And thank you again.
Thank you.